shows some bits.
So this week we're going to be covering the cell cycle. Probably we'll not get all the way through it, and we'll have to finish the rest of it after next Tuesday's exam. So that'll be quiz 3, and then the following, there's actually two lectures left, which gives us enough time to go back and get the mitochondria. Everybody excited about that? So that's what we'll do. And so that's the plan, so you're a little bit aware of that procedure. And so today we have a little bit to finish up on the cytoskeleton, and we'll immediately turn around and use what we know about the cytoskeleton to run the cell cycle. So it all kind of starts to make sense. Everything starts to pull together, particularly the cell cycle. So, okay, so the last little piece, intermediate filaments, we're not going to spend a tremendous amount of time, but remember, they primarily perform a structural role. They're not very dynamic, okay? And we'll look at a little bit of the things that they do provide in terms of structure. And there's no role in cell motility. They're not very dynamic, so that's one reason why it's not a good choice to propel a cell and to do much. But it is a great thing to use to strengthen the cell, to keep them, particularly if they form sheets or muscles or whatever. It's a great way to keep those things from tearing themselves apart when they're distended. You can imagine all the stress that goes into your gastrointestinal system when you eat, all the motility that has to go through there. What is it that keeps that from ripping apart? Actually, sometimes it does, but that's a major mistake when that happens, obviously. Okay, so remember the unique properties. There's a reason they're called intermediate filaments, because they have this broad diameter range, and remember there's a reason why that diameter range is broad. That has to do with the diversity of the filaments themselves. So in this case, remember the rule is the diversity of function comes from the filaments themselves, not from the binding proteins that grab onto them. So that's very different from actin and microtubules. They form a braid-like structure. They're not tubes. They're not beads on a string. No necklace of any kind. It's a computer cable. Okay. They do not bind nucleotides. That's why there's no dynamic stability to them. They're very, very stable. They're the most stable of all the filament types. Okay, so that's the broad characteristics. Let's see if I left anything else out. Yeah, particularly when we look at assembly, you'll see there's no polarity. So that means that if you had a motor, it wouldn't know which way to go, right? All those motors go a particular way, and so they have to have a direction. There is no direction to them. Okay? And so again, all the most diversity of all the elements, there's lots and lots of genes for these different ones. Okay? So let's talk a little bit about assembly and take a quick look at function. Okay. Okay. So yeah, that's probably okay. So this is the epithelial sheet. This is one of our old friends, Mark Simpson haircut. This is the enterocytes. These are absorptive cells. The thing that gives these little deals here are actin. Okay, so that's the reason that shape is there. But notice these little girders that are running across the cell. They're actually attached at these junctions between cells. So these desmosomes are things that are holding on to, these two cells are attached to each other because of the desmosome. And this one up here, that's a tight junction. That actually has a job of being highly selective for letting some things but not everything. A lot of times they're cation selective, or they're anion selective, or they're water selective. Okay? But this thing is sort of a brute. It's permeable, so things can, once they get into this little compartment, this is the basolateral compartment right here. It's essentially in contact with the blood, okay? And so these guys are holding hands. It's not impermeable to anything. It's there for structural support. So if everybody in the room holds hands, this thing, that makes it less likely it's going to fall apart. And the other thing is, you notice, if this thing were to swell, this resists swelling. It resists being pulled apart. So that's what that girder's there for. And then you notice they run from there, they run to this is a hemidesmosome. So when it's not shaking hands with the brother, that's a full desmosome. Okay. The one that's shaking hands with the extracellular matrix, that's what attaches the epithelia to something. It's got attached there. That's the basal lamina. It's got collagen and fibronectin and all this really neat stuff. But it's outside the cell. And so that's the foot grip that the cell uses. And notice, what do? You put a hemidesmosome and then you link these intermediate filaments to stabilize it. Okay? So that's a real obvious structural component. All right, so let's look briefly at how these things come together. Mainly, I don't want you to memorize all this stuff here, but if you just get the overview, you're going to start out with a monomer. You're going to have a coil motif. It's very important in this intermediate filament. Is there a coil motif in any of the other filaments? You got three choices: actin, tubulin, nucleus. This is the only one. Okay, so this is it starts as a coil and it gets even more twisted. Okay, so what happens is you have a coil dimer that's head to head, so that's parallel, and then you take two of those and you arrange them anti-parallel. So this is where it loses its polarity. So this is the N-terminus, that's the N-terminus. That's the C-terminus, that's the C-terminus. So it's anti-parallel, like an anti-parallel beta sheet, okay? Except it's a coil, right? And then you take, so this is the fundamental unit of linearization. It's called the tetramer, okay? And then you stack those two tetramers together, and it forms like a protofilament. And so that makes a protofilament, and then you take 8 of those and twist them into this rod. So that's why it looks like a computer cable. This is really, really strong. Okay. All right, so that's, and no nucleotides involved. So that's the main take-home message. Okay, so what about the diversity? Okay, these are the major classes of the different types. And they go by types. And actually, let's actually go to the PowerPoint. The outline because it says it better. I always prefer what I say compared to the book. Okay, so and it's a little reversed from your outline if you downloaded it the other day, because the way I had it, I had keratins and then I had type 1 and type 2 afterwards. Keratin is not the class. It's type 1 and type 2 are the classes, but they are all both keratins. Okay, and then type 3, that's not a keratin, it's another type of intermediate filament. So both type 1 and type 2 intermediate filaments are both keratins. Okay, and there is dozens of them. Okay, so there's just the cytokeratins that strengthen epithelia, like that Bart Simpson thing, the enterocytes, the intestine. There's 20 different ones, so you can tell which epithelia it comes from. So if a tumor originates in the intestine, colon tumor, it's going to display a specific cytokeratin, different from breast tissue, which is also an epithelial tissue, but it has a different cytokeratin. Okay, so you can tell where the tumor started, which is important because a lot of times they go to the brain, they go to the leg, they go to the liver, and so you want to know where that tumor started, okay, because that'll help you figure out what type of mutations it's picking up. All right, so that's the keratins, and some of them make your hair and your fingernails. That's why they're so strong. They're made out of those cables. Okay. All right, so type 3 rather, goes to a different type of tissues. You've got basically two categories in this type: vimentin and desmin. Vimentin is found in connective tissue. That's also called, developmentally, it's called mesenchymal tissue. Desmin is found specifically in skeletal muscle. Okay, that actually is what encases the sarcomere. Okay, so remember you have sarcomere end to end in. Let's see if I can. If we go to the PowerPoint, I can just speed up there. Let's go take it. Yeah, there. There's muscle. So remember, this is the well here. That's the sarcomere, okay? And so you have there's the the thick filaments, that's the myosin 2s, the stuff in both sides of the golf bag. And then this is the actin filament there, right? Plus, minus, and it's the plus end that attaches to the Z cap. Now, what's not apparent from that is this is a cylinder. Okay, that's a circle. You can't see it because it's longitudinal. But if you were to cut right here and look at it, it's circular. Okay, and that's why the muscle myofibril, which is the contractile part of muscle tissue, looks like that. So, see that myofibril is sarcomere stacked end to end. Okay, why does it have that compact shape? It's because it's lined on the outside with intermediate filaments. Which kind? Desmond. Desmond filaments are supporting that little tube. That's why there's a little space in between these things. So that's how that keeps your muscles really strong. So it's not all about that active binding protein that we talked about, Duchenne's muscular dystrophin. This is also there to stabilize the muscle. Okay. All right, so let's go on. And so we had vimentin and desmin. Neurofilaments. Boy, do you ever need intermediate filaments to help you out with neurons? Think about what a neuron looks like. If you go to the cerebellum, you have all these really bizarre arterizations. They look like trees. The only thing that's going to make that stable is running cables up and down the length of it. Okay? And so one of the types, not all of them, neurofilaments are used to strengthen neurons. And they make the diameter larger, and as we know, if you make the diameter larger, you increase conduction speeds because of the geometry of the membrane surface area. Okay, so neurofilaments, really obvious use for those. Type 5, lamins. This is an old friend of ours that we started studying the day we started looking at the nucleus. This is that key element that's underneath the nuclear envelope. In fact, when you break this up, that's how you break up the nucleus to get microtubules to come attached to the chromosomes. Okay, and how do you get them to fall apart? Well, that's because when you phosphorylate the lamins, there's actually lamin A, B, and C. I forget which one gets phosphorylated, I think it's C. But one of those gets phosphorylated, they let go of each other. Okay? And when they let go, the nuclear envelope falls apart and the chromosomes go away. And so the microtubules can come in and grab onto the what? Kinetochores, right? Okay. All right, so diversity permits us to be able to figure out lots of different things about cells. What else? Okay. And these guys have the fewest binding parts. In fact, I'm only going to give you one. I know you'd be disappointed, but this one's a little confusing because it sounds like something else we've studied. This is plection. Okay? It's like you're plucking think of plucking one of those, good guitar strings or something, and that make you remember what it does. It attaches intermediate filaments to intermediate filaments, intermediate filaments to actin filaments, intermediate filaments to microtubules, and the fourth thing is to the plasma membrane itself. So it's a little busy guy, but that's not the plextrin homology domain, the pH domain, remember that? What does a pH domain allow a protein to do? Plextrin, you put a plextrin, S-T, where's the word here? Yeah, S, plec, and you put a K and then an S, you've got plextrin, okay? Plextrin homology domain allows you to bind to what? It's a type of Lego. What does it plug into? Okay, I'm going to look that up. But don't get those two confused, right? Okay. All right. So just a couple of pictures before we leave this. That's pretty much all the points that I wanted to make out of this group. Yeah, we don't want to do that. Let's go back to, and we will be finished with this stuff. This would go away. All right, there you go. Okay, that's that table. My table is the one I want you to know. And really, all we have here is an example of plectin binding to all these different things. Okay, so I think we should be able to tell. Yeah, so that I'm pretty sure the green stuff, these are immunogold stains, because you're not going to see these unless this is an electron micrograph, okay? You can just see it makes connections between, this is an actin filament, I believe. It's real hard to tell, but it's the thing that connects all that stuff together. And that's exactly what you would expect for something that's there for structural support of everybody. It doesn't need proteins to bind it. It's bound it's bound to itself and the other two fiber types to keep the highway connected together. And that's what it's for. Okay. And so that plectin is the glue that does that. Okay, questions about intermediate filaments? Why is it important? So if I give you questions about distinguishing the three different types, that's the kind of question you're going to see. Okay? So that's the most fundamental thing you need to know is the differences and similarities between the groups. All right, let's do some problems now. Most of these are word problems. They're not really ABCs. I'm going to give you one for just clicker purposes, but okay. I guess it's on the other PowerPoint. So do get your clickers out. Okay. So to help us review a little bit. What must happen to the microtubule for it to stop shrinking and start growing? Okay. What's going to happen? Start with an idea of what it's made out of. That'd be a good place to start. Okay, I tell you what, get with your group and come up with a group answer, and then I'll pick on groups and say, I need to call a call. So get with your group. And you do a couple, do these first two. And then we'll do the second. everybody gets a blank card. Just hang on to it. Okay, all right, so, you ready? You just pick a random number between 1 and 8. Continue to grow. 9. District 9. Where's District 9? You have an alien problem. 9? Here you go. Okay. All right, so what do you think? What has to happen for, let's say it's stopped. What is it, what's the phenomenon called when it stops growing? It actually, does it just gently kind of stop? What does it do? What's that called? Okay. You're an alien, you're District 9. Okay, so that's fantastic. So now, how can you get it to go back in the growing direction? Because you notice these things go like that, they're all over the place. Half-life, 10 minutes. Standard cytoplasmic mitochondria.
It needs to be bound to GTP.
And what needs to be bound to GTP? So what's the fundamental unit that's polymerizing? Is it just a monomer, or is it a dimer, is it a cable, coil?
Each monomer needs to be bound to GTP.
Each monomer, but it doesn't add as a monomer, right? Right. As soon as it's made off the ribosome, and it's dropped in, this is a good test question, it's dropped into the cytoplasm, so it has no signal in sequence, right? And so alpha and beta stick together, high affinity for one another, and they both tend to be bound to GTP. Okay, so that's the only way that can load. Now actin can be bound to ADP or ATP, so there's a lot of differences. But we're talking about this. That's going to load on with the GTP there. Okay, and so what you need that crown of GTP to be with both monomers in the dimer must have GTP all the way around the ring, which is typically what, 13 protofilaments? It can't be just 12 or 10, it's got to be 13, otherwise it'll fall apart. Okay, so that's the key to that question right there. Okay, so the second part, how about group 3? Where's group 3? Okay, good. What do you think? This is like any chemical reaction. What happens if you increase the reactants on this side? Which way does it go? It's called mass action. It goes forward. And so that's going to make it go from shrinking to growing, right? So that's a really straightforward. And this kind of experiment has been done in vitro because they wanted to understand the biochemistry of how the things stick together and all that stuff. Is that what controls growth and contraction in vivo? The nucleation phase, it takes forever and a day, then it goes really fast, it's elongation phase, and then it stops and it reaches some sort of equilibrium at some level of tubular concentration. So concentration is very important, not the cell. What completely controls whether it's there or not there? Synthesis, or synthesis. Yeah. That is what controls polymerization and catastrophe rates. This thing is going to speed up tremendously when we start getting into the cell cycle and go into mitosis. It's going to dramatically change from slow to fast falling apart. What controls that? I'm sorry? Well, it's actually, well, that's part of it. So that's controlling it, but what's actually controlling all of this is the proteins to which it binds. So its binding part of it are the things that cause it to stabilize, the MAPs. That's what I'm trying to get you to say, the MAPs, okay? All right. Now, let's look at these two. Have you already done that? These are fairly, let's just do these. Okay, so what would happen to GDP? They're pretty straightforward, but no GDP were present in solution. it would, not only would it fall apart, it would never build. Okay, so if you just started with a bunch of alpha beta tubular dimers, because you would get the dimers, it's going to form whether or not you got the GTP. It just won't add to the growing polymer if you're missing GTP. On which one? The beta. The alpha is always going to have GTP. Okay. All right, so that's what would happen. There'd be no polymerization. The ones that are there would fall apart. So both of those things are true. Depending on what the initial conditions were, you could say either one of those answers would be correct. What would happen if the solution contained an analog of GTP that could not be split? We have that in our toolkit. What's it called? Pardon me? We have a form of GTP that we've used to cause Dynamin to cause the thing to pinch off a vesicle, but not, it just created a neck because it's a corkscrew, but it's got a split GTP for Dynamin to break off the vesicle. And it just sits there with a long neck on it, okay, with a balloon at the end. That's what you get with something that cannot split GTP. So this cannot split GTP if you put this stuff in there. You know what that's called? That should be, on these little cards, you ought to have a set of cards just for toolkits. Okay? All the toxins we talked about, how to really go through those. Okay? And one of the mistakes that people make when this is just about metacognition, is when you're reviewing those cards, don't forget to review all of them. People will put the ones they know and the ones they don't know, and they'll review the ones they don't know, then they'll get in a test and they forgot to review the ones they know. Because you might not know it as well as you do. So make sure you review all the cards on your last pass through. Okay, it's just a tip. Okay, it may not work for you. What's going to happen? So it's GTP gamma sulfate. There's a sulfate bridging the last phosphate group. GTP gamma sulfate cannot be split because it's got a sulfur atom that's not supposed to be there. And so that cannot be hydrolyzed. Does it look like GTP? Does it smell like GTP? It looks and feels like GTP. So what would that cause that fiber, that microtube? It's going to grow like hell. It's going to grow. And then when it gets to the end of its concentration, that's the only thing that's going to stop it unless a protein binds to it. It's going to freeze. It cannot be broken down because it's got a GTP cap. It cannot be broken. So these are all just very logical if you review that stuff. Okay. All right. So let's go. Let's do a different one. Do this with your group. All right, a few people on this side of the room. Go ahead, Doc. Draw pictures. Yep. Everybody has this question, right? Let me show you a picture that will help. You're trying to figure out what's the the gap for the beta subunit. Does the alpha subunit need a gap? No, it never breaks its GTP. Okay, it doesn't hydrolyze it. It just binds to it. Okay. All right, let's see, where is this? Oh, I'm on the wrong PowerPoint. Let's go up to. Take a look at that. See if that helps. Can you see that okay? Okay, so clearly, so let's get a group. All right, so clearly there's a couple of things you got to eliminate. It's going to have to be a protein that touches it. So that's the first thing. It could be a protein out in solution that comes in and binds, or it could be something intrinsic the way that microtubules cook. Which do you think of those two? Because the gaps that work for RAS, those are somewhere else. They may be attached to the insulin receptor by an SH protein, but they are somewhere else. And think about all the RABs and all those guys, that thing's floating around. The RAS gap and the RAB gaps are floating around. What kind of ribosome are they made on? Free ribosome, right? Free ribosome. Even though they are integral membrane proteins sometimes when they're bound to GTP. So just reviewing for quiz 3. All right, so this is all going to start to fit together, right? Okay, so it's got to have a binding marker. So you got a limited number of choices just looking at that. So this is in a tube before it splits, and it's the splitting of the GTP that gets rid of the cap, the GTP cap. So it's got to be in this form when it splits. So let's touch on the beta subunit. The alpha subunit, right? But then, if that were true, the alpha subunit and the beta subunit start together with the GTP, it split it there, right? So that can't be, it can't just be the alpha beta subunit and the alpha subunit is the gap, right? Because it would split and never come on, right? So let's put it in there and look at this. Now see, this is one array. So the beta is so it's, you know this, the minus n is always where the alpha starts. And so that dark green is the beta. And you always end with the beta, right? Okay, so it could be the fact that two alphas are touching. So that's a possibility. But look at this. Notice that there is a staggered array and betas touch beta. So it's actually three different possibilities. It's two alphas touching a beta. It's betas touching betas, or all of the above. We don't know. We don't know. Okay. But you can do some experiments to find out. But that's the answer to the question, is we don't know. But there's only, I know that's a crappy answer, but that's what science is. Otherwise, you can't have a business. Answer all the questions. All right. Okay. All right, let's go back. I think that's all the, is that all the questions? Yeah, I think we, oh, no, we have a couple more. Let's stay with it. What time? Okay, we can get this in for your question. Oh, okay, this is, oh boy, data. Oh no. All right, so I need for you to look at this and look at the pattern of what's going on here. And I'll have to flip back and forth. There's a couple of, this is, if you've been doing the problem sets, you've done this, you can raise your hand and tell me what the answer is. Okay, because you don't know what the question is yet, but, okay. There is an answer to this, all right? So you have two different experiments going, two different test tubes. What are you measuring? You're measuring viscosity. What do you think determines the viscosity of a fluid that's got active in it? Monomers or polymers? Think about what ARC-2,3 does to a filament, an active filament. What does it do? How does a fisherman catch fish or something into the water? Net work, okay? And so that increases the viscosity. So you can follow the polymerization of active from G-active into F-active by looking at viscosity. So that's a really easy measurement. And you're just going to measure the time course. Those are the things that you need. Okay? So take a look at this, and what's happening is this is the control. It looks like a Michaelis-Menten thing, and this looks like a, this has been inhibited with something. And that's true. But it has nothing to do with the enzyme kinetics, because this is not an enzyme, is it? Or is it? It does have an enzyme, right? It has an ATP, but that's not what determines this, okay? So this is an inhibitor. This is a mushroom thing. This is why don't go out and snark around on mushrooms. Don't do that. Bad idea. I should know exactly what you're doing. Take a mycologist, get John Zach, and get him, take him out there and find those mushrooms. A lot of mushrooms are really expensive, like truffles. And you use a pig to find them underneath the ground. All right, let's get back to active. So you get lots of chemicals. And the reason, why do mushrooms have all these nasty chemicals? They can't run away. They can't run away. They're there. And so they can only use chemical this. You take a mouthful of this, you will die. I promise you will die. And the reason you will die is something that it does to the filament. Okay? And so what do you think is happening to that filament? What's actually happening? So notice the kinetics. There's not just one thing, there's two things, right? So imagine polymerization going on. Okay. What are the possibilities? This is a chemical, so it's got to add somewhere in this filament. It must touch it, right? And there's only one subunit, so it's got to bind to G-active, or it could bind to the filament itself. Okay. So the difference, of course, is in the absence, this is really fast. It goes really fast, and then it stops. Remember, this is in vitro. So what is the determinant of polymerization in vitro? There's no binding partners. The critical concentration. So the concentration of G-active is going to determine where this thing stops. Okay? So they stop at two different places. So surely you didn't screw up the experiment so badly that you put less active in this one. Well, there's the same amount of active in there. Okay? So let's look at it differently. So notice the difference. There's two things. This is slow, and this is fast, and then they end up at some different critical point for some odd reason. Okay? So what's happening? This is the critical concentration. This is not concentration, but I can tell you that's when there's no more growth, what's going on there is the rate of addition equals the rate of removal. It's in a steady state when it plateaus. Steady state. Things are coming off at the same rate at which they go on. And that reaches the steady state slower, right? But it's a different steady state. So something's very different. Now, you say, well, I can't do that. So you're going to use an electron microscope. How can you tell the direction that a fiber is going, okay, using an electron microscope? You can see the active filament, and you know that it has a plus end and a minus end, but you cannot tell which end is which unless you put what into the solution. You get one of Tiger Woods, he didn't do very good. Anyway, playing his clubs, and you really want to piss him off, break off the end of that club, okay? And then you throw that into the test tube, and it happens to be a really tiny bug. Okay? So you put that myosin active binding domain, throw it in there, and it's going to decorate that fiber, right? So what's the shape that it's going to create? Think of the Chevron gas station. There's a reason it's called Chevron. Because it looks like that. Okay. So this is what happens in the absence of cytoclasm. So this is the control, and then this is the other, in the presence of that toxin. And we've actually used this toxin before. You remember that? You will need that on quiz 3 and the final exam is what we're preparing for. Right? It's in your toolkit, so you need to have that stack of cards with that toolkit. That's how you do it. Okay? What does cytoclasm D block? And it's another reason you die. First, you're gonna be nauseated as hell, and then you're gonna get diarrhea, and then you're gonna die. It's really bad. What protein is the blocker? Give me a hint. Somewhere on heart sensor. Glutes, yes. Glutes. Okay. But it does something else. It actually binds to that. So here's the question you're trying to answer. That's what you guys right. Suggest a plausible mechanism to explain how. Well, you read it. This is why I hate these kind of PowerPoints that people show all the time. I want you to sit there and read your PowerPoint. I don't. I'm trying to explain how this thing works. And you have to make the data match the explanation, and vice versa. That's how science works, or the nature of science. If your explanation doesn't match the data, you need to change your explanation, or you did the experiment wrong. That's another possibility. Okay. All right. So what we're looking at, let's cover the book. We have to explain that data. Okay. And that has to match to, remember, no cytoclasm goes fast and it stops higher, goes slow and stops lower. Okay, that's that. And then we have to explain what's going on. This is actually giving you the answer. So talk to your group and come up with, you can do that. Maybe you need to look at your notes. What's the first thing you have to figure out? If you don't know which one is which group, that's the first one. It should be somewhere. That's why. But then explain the other. That's the point, and that's the bar. Arrowhead is point and bar. Let's have the bar. So tell me the first thing is, which way is this going? In other words, not which way, but what's the plus and what's the minus? Remember the rule? I can tell you a really good way to figure that out is not forget it. It's the opposite of what you would think. I mean, that's really what it is. Because you go, which way is this going? Well, the way it usually goes is the plus end goes faster than the minus end. So it usually goes, that's the opposite. This is pointing to that's the negative end, that's the plus end. So that's what, if you just remember that, it's the opposite of which way the gas station thing, the arrowhead is pointing. That's negative and that's positive. And so we know, and the second thing is to remember the rule that the plus end is where polymerization happens and the absence of everything else faster. And look, that's exactly what happens because you get rid of all the myosin, and now this end is not going to be, you can still see it, but it's not going to be decorated. And because you don't need it. You know which is positive, which is minus. And notice, you know that's the plus end, because you know you see the arrow, the bar, that's the point. Okay. And so it's growing faster, right? Is the other end growing? Yes. The other end, is it slower? That's the rule. Okay. The reason this is growing faster than that end is because there's G-actin has ATP. If it had ADP bound to it, unlike microtubules, it'll still add, but both ends will grow at the same rate. So maybe that's what this does, right? Or not. I mean, look at what's different about these two. Yeah. Right? And so it's the minus end is growing. Is the plus end growing? No. Hey. What do you think cytoclasm B has done? It's what? Well, it could inhibit profilin, but profilin, because profilin, that's a good point, but there's no profilin in here. This is a test tube experiment so that all that complicated stuff is simplified. Okay? So you've got two things. You've got cytoclasm B and you've got G-actin in there. Okay? And so look what happens. So this is in the presence. So it's slowed down. So why is that slower? Yeah, it caps the plus. So you shut off one place where it usually grows fast. That's why it's slow. If you shut off the plus end, it can only add to the minus end, and we know that's slow. That's why this slope is less than that slope. Okay. Now, why is the viscosity steady state level? So this is the level at which actin is adding and jumping on. So it's at a steady state there. But that level is lower than that one up there. So the only place that can add and leave is one end. Okay. So adding and leaving and competing at the same end. You don't have two places. This one can add very quickly to this side, and it can fall apart. But it can also add very quickly, a little bit slower. So it's got two choices. One choice there stabilizes at a lower steady state. You can't go fast enough to make that much viscosity. It's a different concentration doesn't change. It's just so slow because you've got on. But here you've got several different means to work with. Okay. So you can get a lot from looking at that kind of data, that's kinetic data, and then there's visual data. And we tend to do this in cell biology, which is what makes it a very powerful field. You can look, you can measure, you can do biochemistry. That's why it's so darn hard to understand all that, because you have to know all of that stuff. And genetics, you know, and Spanish, Japanese, you know. So, all right. So what I want you to do with that card, by the way, so we'll take a break, is I promise you, to rate each other. I want you to rank me, but what I want you to do is slightly different. You need to put your name on it, I'm going to throw it away. All right? As much as you'd like to do an anonymous card, let's not do that. So there's three things I want you to do. tell me one thing you like, that we can keep doing, or you like that. One thing you dislike, and then the third thing maybe that you like for me to do entirely different. Okay. Dislike, like, different. Okay. Just mark them out. Okay, you should go. Okay. Okay. This one. So. This one's true. False. And you. It's hard. It's okay to talk too. Like, it's going to punch me up, isn't it? No. Yeah, we can. If you're on hand, you can hold up your card. Otherwise, you're taking off it for a second. One of the things that will be done differently, the stuff that we're doing right now, what's different about what we're doing now than what we usually do? More questions or more? What about the speed? Slow is slower. Okay. So these kind of things slow the pace down. So how could you do that and get to this maturity? By college we go, you're not going to be able to cover that book. There's two meanings to the word cover, right? There's to go through, and there's to obfuscate, to confuse, to hide. And so covering things too fast and too much is just as bad as not covering. So what's going to happen is, some of you know I'm going to offer this class as an online course, but that's actually going to help me improve the F to F class, which is face-to-face, right? And it's going to be a blended course. And so most of it will be, it's called flipping your lecture, not flipping off, but flipping your lecture. As much as you'd like to do, you're showing great restraint. But so the flipping your lecture is content delivery is done online. Okay? And so that's why I'm walking over to the library every day to that place where you guys got trained composer. And I get in this little room and make a video of myself. It's very irritating because I like to walk around and I sit there. The choreography is really horrible. So if it looks bad, that's why. And so anyway, so that's what I'm doing, recording all the lectures, and that's what you'll look at if you take this class. You'll do that, but you'll have to come to class prepared. And we do problems in class, and then things that people are not getting, I reteach that. So that's how that's going to work. I don't know if I can get there by fall because it takes a lot of work to develop those problems. But that's where I think most professors should go. How long do you think that's going to happen? Take that to happen for most of the people that do that? Probably never. Actually, it takes a lot of work. So, okay. Take a quick break on you. And just hang on to your cards, actually. You can just throw them up here when you leave, because you might think of something else. Tie doesn't matter. Okay. Okay, we're going to start one of my favorite topics. And one of the things I like about this is it integrates everything we talked about, with the exception of transport, which is sort of a peripheral issue. But the cell cycle, that's exactly what it should do because it's the life cycle of the cell. And it's pretty straightforward, but there's some certain things that we have to get straight about it. So what we'll do is, there it is. It's the cycle that a cell goes through in order to divide, get bigger, duplicate its DNA. So you start out here mitosis, and I don't know what that's about. So that you split off two daughter cells. That's actually separate from, it's in M phase, but mitosis is not the same thing as cytokinesis. These are two different processes. And you know now what causes cytokinesis. It's a little mini sarcomere that goes around and squeezes the belly between the two Siamese twins, and they come apart, right? And so you go from this is always the shortest phase. That's usually, if this is a 24-hour cell cycle, we do that just to make people think about it like a clock. This is like 30 minutes. It's very fast. Okay? This thing is very slow. This is G1 because it has to make a few decisions. And it's kind of like going through old Europe, when you went from East Germany to West Germany, you had to go through checkpoints. And so that's what you have to do here. There's actually 3 different checkpoints to go through. So you go from G1, you go into S, and S takes a little longer than mitosis, but it's pretty fast. But so it's probably a little bit slower than G2 and G1. This is S phase. You have to be pretty careful though. This is you're duplicating your DNA and your centrosomes. That is your centrioles. Why? Because you got to have 2 poles, right? Those are the intox, the 2 poles. Okay? And then you're going to go in here, and that's some of this is you can think of. It stands for gap, but you can think of it as growth. You actually have to reach a certain cell size before the cell goes, I'm big enough to divide. Now, the hallmark of a cancer cell is number 1, it stays in here all the time, but it doesn't give a hooey about how big it gets. And so that's why people like Tina Gumney, who works on a classic system like C. elegans, is interested in talking to people in the medical school. They may hire her actually in cell biology because they study the effect of cell size on staying in and out of the cell cycle. So it's a really big relevant topic to cancer cells. So the cancer cell does not care about its size. So guess how you can always tell a cancer cell from a regular cell? Tiny. And so you look at a tissue, histological sample, and you look at liver, and you see all these little dots. They have no cytoplasm. The nucleus is about as big as the cytoplasm. It's just a little ring of cytoplasm because it didn't wait. It just kind of went, I don't give a heck. And it went on into, duplicated its DNA, and it didn't do that very well because it doesn't care. It wants mutations. This is very fast evolution. The more mutations, the better. Okay? And then it doesn't wait there. It goes right in here, and it just goes over and over and over. Now, a normal cell can make two types of decisions. It can stay in the cell cycle as long as it wants to divide, but then it will exit the cell cycle. And there's only one place it can exit without dying, and that's G1. Okay? Why would you want to exit the cell cycle? Well, if you've got to grow, you've got to have two different things. You've got to have enough nutrients. You got to make nucleotides, and that's where your transport actually comes in. So you got to have adequate nucleotides and adequate glucose and amino acids, because when you grow, you've got to build protein and you've got to do all that stuff. The second thing is, in order to get past these checkpoints, you're going to need some growth factor, like insulin or whatever. And if we can get to it, we will get to it, because I have the time this year, is they control whether or not you stay alive. There's two things. You'll either exit the cell cycle if you have no growth factors, or you'll commit suicide, apoptosis. Right? And those two things are prevented by having growth factors. You got to have that. And there's a reason why that prevents cell suicide, programmed cell death is the kind of word, okay? And keeps you in the cell cycle. Okay, where do you go? So now cells, that's one reason that you can exit, because you're starving and you don't have growth factors. So the second reason that's good is if you differentiate. So you can't do both. You can't decide to be a retinal cell and then keep dividing. You can be a retinal stem cell and stay in the cell cycle, but you can't differentiate into a retinal neuron and keep dividing. You would have to de-differentiate, and you can do that. It's difficult. It doesn't happen a lot in the nervous system. You'd have to de-differentiate to come back to G1 and then re-enter. So differentiation is exited to, this is called G0. Imagine this little blue down here. I think I might have one that has that blue. G0 is where you go to exit the cell cycle and stay alive. Okay. If you stop in the cell cycle, you've got a certain amount of time to fix your wagon or you're going to die. Okay? Your cell has a mechanism to know if you're stunted and you're not going anywhere or you're mutated. That's the way it's supposed to go. You're supposed to quietly and very orderly chew yourself up. That's called apoptosis. Okay? All right, so that's what you want to do. And so you're asking yourselves these questions at 3 different checkpoints. So there's a checkpoint here that's called restriction in mammalian cells. It also has a special name. It's called start in yeast. And the reason we almost retain that yeast terminology is we had no idea of how our own cells did this. There were 3 different types of model systems that allowed us to figure this out. And the reason it worked is that this is so fundamental from the beginning of cells that it's the same in sea urchins, yeast, and the frog Xenopus. Those were the 3 systems. Not one of those systems had all the answers. The sea urchin has this one, and then the frog had this one. And then they suddenly all went to a meeting and they just went, and they talked, and all those ideas came together very quickly. And then the medical people, then they picked up because they knew they were drugs, didn't they? Yeah. All right. So then we got our answers from those model systems. So that's why Drosophila and C. elegans and these things are really important, because we can figure out really basic stuff. And nobody cares if you cut up a worm or pull the wings off of a fly or put drugs on the yeast. They just don't, PETA is not going to come after you. It's the people who can taste the animals. So anyway, so this is restriction, okay? And then you go to the second one. This is the G2M checkpoint, or transition. We also call this NE, mitotic entry, because the checkpoint, this is a really important one, because if you got to have all your things in a row, or you're going to be really messed up, and you will never get through mitosis, because the cell does not want you mutating unless you're a cancer cell and you can't control yourself. Okay, so you go through here, you enter mitosis, and then you go very quickly and you stop again at metaphase. That's the 3rd checkpoint. So this is the metaphase to anaphase transition. Because you can imagine, what are you really looking for? All your chromosomes are lined up. What are you asking yourself before you pull apart into 2 daughter cells? What's the one thing that must be true? How do you leave a chromosome behind? that happens.
It's not attached to the genetic cord.
That's right. Your microtubules are not attached. So the cell has a really elegant way of figuring that out. It has to do with two guys called Matt and Bum. Okay, we'll meet those. Maybe not today. But, hold on, we got about 11 minutes. All right, so those are the 3 checkpoints. So there's 2 things that you have to get past the checkpoint. It's almost like going from East Germany to West Germany, okay, which is a lot better now. You need a car, and so you need an engine. And so the cell cycle engine is a kinase. Surprise. Everything, notice the signal transduction, seems to be controlled by the enzyme. And this is a serine threonine kinase, okay? And it's a heterodimer that depends on another protein called the cyclin. So you can think of it as like a bicycle with an engine, right? And you got to have both of those stuck together. That's called the cell cycle engine, is that kinase. And you need different kinases generally, but not always, to go through these 2 cells. You have to have the right kinase activated. And then, this is the weird one, to get past this checkpoint, you have to destroy the kinase. So, activate, destroy. Okay. And the same kinase runs this one and this one. Okay. And so this, you can call that ME and that MX, mitotic exit. That's quicker than writing down all that garbage, right? Okay, the second thing is you need the engine and the bicycle stuck together, active kinase, serine threonine. You need to have completed all of the steps since the last checkpoint. Okay, so if you're going to go into mitosis, what is it that you need to have finished since this last checkpoint? So the rule is you go back to the previous checkpoint, and you must have completed all of these events. Those are called the downstream events. What are they downstream from? You can only go, this is really important, and there's a reason for this. It's unidirectional. I mean, it's like a clock. It doesn't go backwards. It goes this way, clockwise. Okay? So you cannot go backwards. So it's like a river, and so there's upstream and downstream. So it's important to understand that distinction. There's a directionality. And so what you do is, all right, I need to get through this checkpoint, so I know I need an active kinase, cyclin-dependent kinase complex, CDK for short, CDKs. All right? But I need to go back to this and ask myself, have I completed all the stuff downstream from the last checkpoint? So I want to go through that one, so I go back, and I go, have I duplicated my DNA? Do I now have a set of 4 centrioles? Because remember, pole takes 2 Yankee for perpendicular centrioles. So now I don't need 2, I need 4. Okay, that's the poles. Have I duplicated those? I've got the duplications, but did I create any mutations? Is any of my DNA damaged? You've got to repair it. So that's the stuff you've got to complete before you get back in, right? So what would this one be? So you want to go through this checkpoint. So as I mentioned, you got to destroy the kinase. That's what makes this unidirectional, by the way. When you destroy something, it means you can't go backwards, right? So you got to destroy the kinase, and the same rule applies. You go, this is the previous checkpoint. What are the events downstream from this checkpoint? What are they? This is metaphase, this is prophase, right? I mean, this is going into prophase, so you have to have nuclear envelope breakdown. That allows the microtubules to come in. The microtubules have to become incredibly unstable, but stable in a certain way to make the microtubule, make the spindle fibers. So you got to have spindle fibers, they got attached to the chromosomes. All of that is downstream from this checkpoint here, right? You're going through this one, you go back to the previous one and ask what happens in between here. All that has to happen properly. And it's checking itself. There's little molecular ways to check yourself out. Those are the 2 things. It's always about activating and deactivating the kinase, and then did I do everything right to get here? Because if I messed up, I want to stop and repair, or my other alternative is if I can't, I undergo apoptosis. Okay. So that's a pretty straightforward rule. Okay, so here's the engine, cell cycle engine, and this is the real engine. That's the kinase part, CDK, cyclin-dependent kinase, and this is the cyclin. Okay, and the reason it has that name is it turned out the first thing they noticed about this protein in one of those, I think it might have been yeast. They go, the kinase doesn't change. It's almost always there, except when it's being destroyed. But it usually doesn't get destroyed. It's the cycling. And so it cycles up and down. So this is staying there, but without its binding partner, it doesn't work. Okay? So what they noticed, well, there's all these different cyclins. And we're not going to memorize all this list, so we'll really call them a G1 CDK or a G1S CDK. You need both of these to get through restriction. And S CDKs to get through S phase, and this one is the one we're going to focus on. And I will ask you to remember its particular name. So it's CDK1 and cyclin B. That's the only one you have to memorize. You need to know these, but not all the components. Right? These are all yeast, and this is vertebrate stuff. Okay? So we're going to really focus in and use this as our model. Cyclin B activates CDK1. And it's called the mitotic CDK. It's a good name for it, right? Okay? Now, so this is what they found is, the kinases, CDKs, were there all the time, okay? But what was happening, see, this is the, let me see how this is done. S-cyclin, so that's this is the cyclins up here. They were going up and down. This one is kind of blobby because you need it for quite a while. Get through actually G2 and S. So it's not important to memorize the patterns except to see that there is a pattern. There was a little lump, and then there was this blob, and then there was, this is the one that we're really focusing on, is this M cyclin. And so it would start to go up at G2. This is the one you gotta have to go through into mitosis. So the G2 mitosis transition, and a quick way to say that is mitotic entry channel. All right? So that's the one. So the kinase is there, but it cannot be turned on. Why? Why do you need a cyclin? That's the next question you can answer. Okay? All right, so we're asking ourselves different questions. If this one, we have a few more questions to ask than normal, because if you did your business correctly, you stored up nutrients and you got all, nucleotides, amino acids, and all that stuff. What else did I say you needed? You got to have your nutrients, weightlifting. Then you have to have growth factors, insulin. You got to have epidermal growth factor, and all that stuff. Okay. So is the environment favorable? And size. Those are the three things. You got to have this right size. How does size detect anything? Concentration. All right. You got to have enough number. Okay. Okay, and so different things throw a wrench into different things, okay? And so we're going to pay a lot of attention to this right here. And this is all about, did the downstream events of the previous checkpoint, have they been completed? So is my DNA replicated? If it's unreplicated, I freeze, okay? And you can repair that, all right? If not, you off yourself, okay? Or you sit there until it's repaired. DNA damage, okay, so you're out in the sun this summer or tomorrow or whatever today, and you're getting UV from the sunlight, it's damaging your DNA, and when it does that, irreparably, and it cannot be repaired, that cell will kill itself, so that you do not develop a carcinoma. So that's a good thing. And it's slightly different, so DNA damage is a really nasty thing, and so it puts a block on several of these. But these two I like as an example because it makes a lot of sense. You got to duplicate your DNA, and it better not be messed up, unless you're a cancer cell, and then you don't care. One eyebrow, one leg. It's all it doesn't matter. Okay, and this is the guy that kills the kinase. So we'll get to that. All right. So briefly, let's just talk about the big picture of the engine for a second. And what's going to happen is, the CDK is inactive because it must have the binding partner. There must be a reason. I mean, they don't like each other. The reason is this loop. This is called the T loop because it has a very conserved threonine residue, and it is in the way when there's no cyclin. The binding of cyclin will move the T loop out of the way, partially. So actually, cyclin activates it, but it's only partly active, it's weakly active. And there's actually a good reason that you want it sort of weakly to start out with. All right, and then what happens is There are certain activating phosphates that have to be added, and there are inhibitory phosphates that need to be added. Okay? And so phosphorylation will control both inhibition and stimulation, just to confuse cell biology students. That's the reason involved in it. Make it hard on your life, right? But it's actually a really straightforward story, okay? And that I can't tell you in 1 minute, so we will get to that. Next time on Thursday, then of course the test is a week from today. I didn't post the answers to the practice test.